All right, I am very pleased to introduce today's seminar speaker, Dr. Isabelle Cote. Dr. Cote is a scientific diver, research chair, and professor of marine ecology at Simon Fraser University. Her work focuses largely on behavioral ecology, coral reef ecology, and conservation, with a particular focus on subtidal research. Dr. Cote, thank you so much for agreeing to speak today. I will turn off my camera and hand it over to you.
Great, thank you very much, Mary. Thank you for the invitation to speak today and for the nice introduction. So what I'm going to try to do today is summarize in sort of one big story the last 10 to 12 years of research that I've done on one particular alien that has found its way to the Western Atlantic. So I call this a story because it's very much what it is. I mean, nothing was planned at the start, and it's sort of developed, and we're at the point now where we know a lot about the system and we can make sense of it. So what I'd like to start with is just tell you a little bit about invasions in the marine realm. Then I'll talk about the star of the show, which is the lionfish. I'll tell you what we've discovered over time. So we focused a lot on direct impacts and implications for management. Then we'll go into broader consequences, so indirect impacts, and you'll see some of the avenues in which this research program has taken me, which I never could have predicted right at the start. And I'll spend the last minute or so talking about what the future might hold. I'm quite happy to be interrupted at any point in time if you have some pressing questions. So I think it's fair to say that marine invaders are found everywhere in coastal areas. This is a map from a study that was done some time ago now that shows the number of known harmful alien species in various coastal regions of the world. And what you can see is that pretty much all the coasts have some sort of color, some more intense than others. The bits that you see in white are not places where there are no harmful alien species, but places where we have no data for. So it's pretty, I'm pretty certain that if we got data from these places, we would find some sort of introduced species there. But the kinds of marine invaders I'm going to talk about today are really quite rare. So these are predatory marine fish that have become invasive. And in fact, you can probably count the number that there are on the fingers of one hand. And most of them right now are found in Hawaii. So about 60 to 70 years ago, fisheries managers in Hawaii decided that there was an empty niche of mesopredators because there are very few native groupers in Hawaii. So they very much on purpose introduced groupers and snappers, two kinds of tropical mesopredators from other places in the Pacific. And nowadays, these peacock groupers and blue line snappers form the majority of the biomass of mesopredators that you find on these reefs. So that picture of a blue line snapper that you see here is a picture that I took when I went on holiday in Hawaii. About 10 years ago, people started becoming interested in finding out what the ecological impact of these species had been. But I would argue that these studies really have come, 50 years too late, because any impact that they would have had would probably have been disappeared. And there's a lack of baseline data to compare from pre-invasion times. Now, this problem is not one that we have for lionfish, which is arguably the best documented marine invasion in the world. So when I talk about lionfish, I'm in fact probably talking about a hybrid between red lionfish, which has its native range throughout the Pacific, and common lionfish, which is found in the Indian Ocean. So where these two species meet throughout the coral triangle is where they hybridize, and that has been the source of the aquarium trade that has brought lionfish from the Indo-Pacific to the Atlantic. So the first sighting of lionfish in the wild was off the coast of Florida in the mid-80s. So you can see the dates are scrolling. And for about more than a decade, lionfish did nothing. Then they jumped to Bermuda in the early 2000s, went up the eastern seaboard of the US. 2004, they made it to the Bahamas. And after that, they spread really rapidly around the Caribbean Sea and into the Gulf of Mexico following prevailing currents. Right now, in 2020, they have invaded basically everywhere in the Caribbean Sea and the Gulf of Mexico. They have been found down the coast of Brazil, and a couple of weeks ago, one has even been caught in England, believe it or not. So this represents really a rapid invasion, particularly since 2004, when they made the jump to the Bahamas. So lionfish are now found pretty much in every habitat. So they're found on coral reefs, which is where most of the studies have been done. They're found in mangroves. They're found in seagrass. They're basically found anywhere where you have some sort of structure that gives them shelter. They're found as shallow as 1 meter deep, down to more than 300 meter deep. So they are a generalist, really, in terms of habitat. And I always say that if you were to devise the perfect marine invader, you probably would design something that's a lot like a lionfish. So you have a generalist predator that eats things that are up to half of its size, and a fish that's relatively large. So that means a large range of potential prey. You have a coloration that to us looks very kind of gaudy and glamorous, but in fact, in most of the habitats where this species is found, makes the fish quite cryptic. You've got a species that has venomous spines on the dorsal and ventral fins, which means they have very few native predators. Under optimal conditions, females breed year-round. They release buoyant eggs that are enveloped in a jelly mass that some people actually think is not palatable. They have a long pelagic larval duration that allows the larvae to disperse very far from their spawning location. And finally, they have a very unusual hunting mode, which I'll show you in a second. And this hunting mode is like nothing that native fish in the Western Atlantic have ever experienced before. So our story begins in 2008, about 4 years after the first lionfish arrived in the Bahamas, but there was very little awareness of this. So I sent a student to the Bahamas ostensibly to work on sharks, and she got there and emailed me and said, you really should come down here because the reefs are crawling with lionfish. And I thought at the time, where have I actually sent you? I thought you were going to the Bahamas. There's no lionfish in the Bahamas. And then she sent me this picture that had been taken by a diver, and I went, holy cow. So I went down to the Bahamas to look at things for myself and realized there was an amazing opportunity here to study an incipient invasion and see what the impacts were. So the first thing we did was to figure out how, basically how abundant and big they were. At the same time, I had a student who was working in Kenya in the native range of lionfish. So I got her and my student in the Bahamas to do exactly the same kinds of surveys to compare the density, length, and weight of lionfish. And you see the results here. So in terms of density, what we found in 2008 was about 5 times the density that lionfish were found in Kenya. In the Bahamas, they were about 50 times bigger on average and weighed 3 times as much. So that means that in terms of biomass, what we saw in the Bahamas was about 13 times higher than what my students saw in Kenya in the native range. So this is really the hallmark of an invasive species. So I mentioned the unusual hunting mode. So this little video here is going to show you exactly what they do. So this was taken in an aquarium. You can see there's a little globy that's been thrown in there, not for my benefit, but I was there, so I recorded it. And you have a young lionfish that's going to show you exactly how a lionfish does it. So you can see that it's adopted this very stereotyped pose with the head down and the tail up, staring intently at the prey. And the thing obviously that you've noticed is that the lionfish has very large feathery pectoral fins that it is now holding sort of at right angles to the body, essentially creating a curtain that prevents the prey from moving in that direction. In the wild, they can hold that pose for 5 or 7 minutes. They're incredibly patient. And they often sort of slowly move the prey towards a vertical surface, effectively blocking all of the escape routes. So the fish is about to make one mistake, and I'm going to stop talking because it's about to happen. Don't blink because you might miss it. There you go. Okay, so lightning speed, this kind of thing is happening millions of times everywhere on Caribbean reefs right now. So for anybody who's ever spent time watching animals, any animals, you probably know that witnessing predation is an extremely rare event, except when you're watching lionfish. So we spent a lot of time early on just watching lionfish during the daytime, which is it's not even when they prefer to hunt, but in the daytime we could see them hunt. We were able to measure a predation success, so more or less 3 times out of 4 they were successful in eating the thing they were targeting. We were able to measure a predation rate, a daytime predation rate of about 1.5 prey per fish per hour, which extrapolated means a whole lot of prey. We also discovered that the conditions during the day, so how overcast it is, makes a really big difference. So when it's overcast, mimicking conditions at dawn and dusk, when they do prefer to hunt, they spend a lot more time hunting. They move a lot more than when it's sunny. In the early days, we also spent a lot of time catching lionfish and dissecting them to see what was in their stomach, to understand what it was that they were eating. And we quickly came to the conclusion that they seemed to be eating virtually everything. So this is pretty typical of what you see in a lionfish stomach. So these are all little native fish. Some of them we could identify to species, but some of them were too digested to be able to be identifiable. So we really needed a method to identify the unidentifiable. So that made me wish for a little while that we'd have one of these things. Usually when I give that talk like face-to-face, I ask people where they know, if they know what that thing is. I'm just going to give you the punchline. This is a tricorder that Mr. Spock on Star Trek used to bring down to these new planets to figure out what the new species of plants and so on were. So tricorder is exactly what we needed to be able to shove a little unidentified bit of fish to figure out what it is. But, you know, DNA barcoding is just as good as a tricorder. So in DNA barcoding, we sequence a little bit of one specific gene that is really conserved within species, but really different between species. So as long as that sequence has been linked in a data bank to a particular species, we're then able to identify our unidentifiable species through barcodes. And that turned out to be a really, really effective method. So this sort of egg here represents the diet of lionfish as we knew it before barcoding from a very large study of more than 1,000 lionfish in the Bahamas, where the prey in the stomach were just identified visually. So we went to the same place, collected only 10% as many lionfish, used visual identification as well as barcoding on prey that we couldn't identify. And in our 130 lionfish, we found 37 native fish species that have been preyed upon. There was a lot of overlap with the other big visual study, but we found 16 species that hadn't been identified visually. Okay, so the number of species that they prey on has increased a lot since then. Certainly our knowledge has increased, but from our study, we had the first record of cannibalism in lionfish. So we found evidence that lionfish, large lionfish, eat small lionfish. But the hopes that we might have had that this, lionfish could have provided a solution to the lionfish problem were short-lived, because very few lionfish are in fact cannibalistic. So we found less than 5% of the population of our sample had eaten other lionfish. So having a complete list now of the fish species that they like, we were in a position to determine what lionfish like best. So with Stephanie Green, whom I'll talk more about in just a second, we did a study at 2 spatial scales of lionfish diet. So we did what's shown on the top there, these individual-based studies where we watched lionfish, what they ate, and then we did a survey immediately around them of what was available to them. And then we added to this a large-scale survey of reefs where we did transects, counted all the lionfish, and then counted all the small fish that were available to them at the reef-wide scale. Now, interestingly, at these 2 really different scales, the same characteristics of prey turned out to be important for their risk of predation. So prey that were small, that were cigar-shaped, that were solitary, living on or near the bottom, that weren't cleaner fish, and that were nocturnal. All these characteristics were associated with a higher risk of being prey to lionfish. And it turns out that there are really many species on Caribbean coral reefs that have several of these characteristics. So these 3 are just an example. The top one is a juvenile parrotfish, which, in addition to having most of these characteristics, is also ecologically very important because they graze on algae and maintain a balance between algae and corals. So all in all, the species that have all 6 of these characteristics, we figured, were about 200 times more likely to get eaten by a lionfish than species that had the opposite characteristic. So I think we understand what lionfish go for really well now. How about their impacts? Can we measure direct impacts? So this was the main theme of Stephanie Green's PhD. She was the first student that I put onto the lionfish system. So Stephanie first started working in New Providence in the Bahamas, surveying a bunch of reef in the southwest of the island. And the main question that we asked here was how unsustainable lionfish predation is. And we used a really simple approach to address this question. We figured that we probably could quantify prey fish production, so the rate at which prey fish are producing and replacing themselves. And we could definitely measure from behavioral observations and so on lionfish prey consumption. Now, the difference between those two is net prey fish production. So predation should be considered then sustainable when that net prey fish production is 0 or positive. Now, I'm going to spare you the details of the modeling that went into figuring out what prey fish production was and lionfish prey consumption. It involved a lot of data, some assumptions as well, but the bottom line is this. So what you see here are a bunch of sites along the bottom, different reef sites, and you've got net prey production, so that N on the y-axis. So the horizontal line at 0 is where prey production and lionfish prey consumption balance each other out exactly. So you can see there's only 2 sites at the extreme right hand where N is near 0 or positive. That means that all the other sites are sites where lionfish are consuming native fish faster than the native fish can replace themselves. And just to give you and sort of give you some context for these numbers, that reef right here would need to have twice the native prey production that it currently has to match lionfish prey consumption. This reef over here would need to have 4 times prey production, and the reef that is in the worst shape would need to have 12 times a higher prey production to match lionfish consumption. Now that looks like a really grim picture, but it is modeled. And if this model is right though, we should be seeing a really big decline in native prey fish on these reefs. So what Stephanie did at the start of her PhD, she measured prey biomass for 42 species of prey fish that we had identified in lionfish stomachs. And then she redid the same surveys on the same reefs using the same methods a couple of years later. And this is what she found. On every single reef, there was a decline in prey biomass. And when you put all of these reefs together, it amounted to an average decline of about 65%. Now, this is something that actually would not have been picked up in conventional surveys that focus generally on fish that are bigger than 10 to 15 centimeters. So these are little fish that usually, up until then, nobody really cared about. Okay, but this is a massive, massive decline. So we clearly have a problem and we need to deal with the lionfish problem. Now, early on, some people thought that maybe groupers, native groupers, would be the solution. So these are mesopredators that can grow to quite a large size, more than a meter. And that shark student, if you remember from the very beginning, continued her work on sharks, which entailed looking at the prey of sharks, groupers. And when she caught groupers, she looked into their stomachs. And lo and behold, 2 of the groupers that she found had juvenile lionfish in their stomachs. So she published a little note about this. And that really raised hope that groupers could get us out of this mess. This was followed a couple years later by a study, again done in the Bahamas, that suggested that maybe groupers were indeed the solution, because in and out of marine protected areas where these authors had surveyed fish, they found a negative relationship between grouper biomass and lionfish biomass. So where there were lots of groupers, there are high grouper biomass, there was low lionfish biomass. Now that sounded quite promising. The only problem is that that didn't seem to jive with what I was observing at my sites in the Bahamas or what some of my colleagues were observing elsewhere in the Caribbean. So all these colleagues and I decided to pool our data to see whether this relationship held at a larger regional scale, and unfortunately it didn't. So it doesn't seem that groupers are the solution. And at that point, another PhD student of mine, Nicola, became interested in finding out whether that what seemed now to be a fluke of finding two groupers that had lionfish in their stomachs out of 7 groupers that had been looked at, whether that was just a fluke or really there was something going on there. So Nicola Smith, whom you can see here, worked in the same place in the Bahamas and did the largest survey of grouper stomachs, working with fishers in the region. And she found a grand total of 0 lionfish in the stomachs of these groupers. So groupers are definitely not the solution when it comes to direct predation. But Nicola was also interested in the potential for groupers to perhaps provide some indirect pressure on lionfish. So she ended up working in another place in the Bahamas, in Eleuthera, on patch reefs that varied naturally in grouper density, and that's what you see on the x-axis here. And she looked at the behavior of the lionfish that were on these various patch reefs. And one thing that jumped out to her was that the likelihood that the lionfish was hiding at first sighting when she did her surveys varied with grouper density. And you can see the shape of that relationship here. When you have high grouper density, it's almost always the case, with a probability of 1, that the lionfish are going to be hiding somewhere deep in the reef, which would be something that's consistent with the perception of a high predation risk on these reefs. When groupers are far less abundant, lionfish are more likely to be out. And what they're doing at dawn, when these surveys were done, is that they're hunting. So hiding is incompatible with hunting. And so that suggests that on reefs where there's lots of groupers, the success rate or the predation rate of lionfish might be a bit lower. Okay, so groupers might be a tiny part of the solution, but indirectly. But clearly, we need a different solution, and the cows have an idea, which is to eat more lionfish, so remove them from reefs. Now, we're usually really, really good at, fishing out populations. We've devised a whole heap of methods that drive fish populations down to near extinction very effectively, but most of these methods actually don't work with lionfish. So people have tried to trap them. They swim into traps, but they swim out of them. And you can't easily use any kind of bottom net because of the habitat, the very structured and hard habitat where they live. So really the only way to get rid of lionfish is to go after them by spearing. So getting them one by one, which from a management perspective is a bit of a headache because that's a really labor-intensive method. Nevertheless, Stephanie and I decided to try to provide advice to managers on how many lionfish they should remove from individual patches of reef. So we thought we would go back to the simple N, P, and C equation and use that as a means to provide advice. So with sort of ecological impacts in mind, we thought that what we're trying to do here is to get N to 0 or to keep it positive, but certainly not negative. Okay, so we set N at 0. We know we can measure P because we've done it before. And now it's a matter of figuring out what consumption by lionfish should be to get us to an N of 0. Okay, and because consumption is directly related to the number of lionfish, we can use this equation to make a recommendation for managers. So what Stephanie did at this point is she undertook a 2-year-long experiment to see whether the predictions from this simple model would actually come true. So can we use this to make recommendations that will actually work? So Stephanie moved her operation to Eleuthera, where she did this massive field experiment on these patch reefs, which were divided into 4 different treatments. So in red, you've got the patches where we did monthly full removals of all the lionfish. In white, no removals were ever done over the 2-year period. And then you've got the intermediate 2 treatments that were guided by the model. So in yellow, we had patches where the model had predicted a certain number that would get us to 0 in terms of N, and we removed a number of lionfish that was likely to be enough to get us there. And then the patches in blue, we removed not quite as many, so we didn't expect to get to an N of 0. So we did these removals and maintaining these densities every month for 2 years. And it's amazing that a 2-year-long experiment can effectively be summarized with 4 data points, but it can. So what you're about to see here is the proportion of change in prey biomass from the start of the experiment on the y-axis. So 0 means no change. That's effectively an N of 0. On the left-hand side, you've got the 2 treatments that our model predicted would be sufficient to prevent any impact. So if it worked, we would expect the averages here to be around 0 or positive. And then on the right, we have the 2 treatments where our model predicted that the removals would be insufficient to prevent impact. So we expected those numbers to be negative. And this is what we got. Sometimes ecology is kind of neat. So clearly, the insufficient numbers that were predicted by the model, that part definitely worked. We continued to lose native prey biomass when we did not remove enough lionfish. Looking on the other side, on average, we just about succeeded. There was a lot of variation between reef patches, which is makes it a little bit difficult then in terms of providing exact recommendations to managers. But the one thing that really surprised us and in a way delighted us is that the success of the most lionfish removed and the all lionfish removed was about the same in terms of maintaining native prey biomass. And the reason why we were happy about this is because it took about 30% less effort to leave a few lionfish behind than to try to remove all of them. So from a management perspective, that's a really important thing to know. But of course, that led to the question, why is it so hard to catch these last few lionfish? So we designed an experiment to try to figure it out. So we used these same patches that were in that 2-year-long experiment. So we used the patches where most of the lionfish were removed, and we used patches where no lionfish were removed. And we did these approach experiments where a diver would approach very slowly a lionfish. The diver was actually carrying a spear like they would when they were culling the lionfish. And as soon as the lionfish exhibited an alert behavior, which was usually to raise their venomous dorsal spines, the diver stopped and another diver came in to measure the distance between the lionfish and the diver. So we call this the alert distance. And what we found was that the lionfish that were living on patches that had been culled every month had a longer alert distance than the lionfish living on patches that were never culled. So they became alert at a larger distance from lionfish. Now, we don't actually think this is a learned response. What we think happened is that the divers, when they were culling, they were probably culling the bold lionfish first. So what was left behind were the shy lionfish. And in fact, another type of data that we collected in this experiment seemed to support this. Every time when we surveyed these lionfish and did these approach experiments, we scored every individual lionfish on these patches depending on how deep into the reef they were. So 1, as you can see here in the picture, is fully exposed. 2 is visible, potentially killable with a spear, but 3 is very well hidden and not possible to kill. And you can see that on average, the median hiding score for lionfish that lived on culled reefs was significantly higher than it was for lionfish from unculled patches. So culling seems to leave shy lionfish behind. So this culling regime, which is effective in terms of reducing lionfish numbers, is also changing the behavior of lionfish, making it harder to cull over time. Now, Nicola was also interested, sort of being from the Bahamas herself, and knowing that a monthly culling regime was not something that would be sustainable for managers in a country where budgets and person power are limited. She did another 2-year experiment to look at whether more infrequent culling would also work. So we see a shift in the type of analysis here with generalized linear models. So here what you have are parameter estimates. Negative parameter estimates mean a reduction in lionfish density, positive mean an increase. And you can see the effect of quarterly culls and biannual culls compared to no culling at all. So these infrequent culls were actually quite effective in terms of reducing lionfish numbers compared to no culling. This is a great thing to know, that you don't have to be out there every month, but you can be out there much more infrequently. However, how many times do you think you can run a 2-year experiment without something happening in that 2-year span? So nothing happened during Stephanie's 2-year experiment, but Nicola's 2-year experiment was interrupted right in the middle by a hurricane hitting the Bahamas straight on. Okay. So Nicola decided to put hurricane as a factor. So pre and several months post-hurricane versus the period of impact of the hurricane. So the period of impact of hurricane on the whole reduced lionfish numbers compared to before and well after the hurricane. But the really interesting thing was that there was an interaction between culling and the hurricane period. And that interaction created higher lionfish densities, which again is something that's not very convenient for managers. So what we think happened is that the hurricane displaced a lot of lionfish. Think of lionfish in a giant sort of washing machine. And then lionfish redistributed themselves with many of them colonizing our culled patch. And the findings of another of my PhD students, Natasha Tamburello, gives us some insights for why that happened. So Natasha's PhD was on the movements of lionfish. She spent a lot of time tagging lionfish in different habitats and following them around. And what you see here is the distribution of movement of lionfish that live on patch reefs. So the type of habitat where Nicola's experiment was done. And you can see that the majority of lionfish are homebodies. They really like their patch. They tend to stay there for months on end. They don't move. But when they do move, they move a pretty long way. And what Natasha found out of looking at these movers is that when lionfish do move, they invariably move from areas of high lionfish densities to areas of low lionfish density. So what our experimental culling did was to create these sinks for lionfish movement. So all these lionfish that had moved around ended up moving along that gradient and finding our culled patches, which had low lionfish density. So that kind of wraps up the cohesive, let's say, lionfish studies. And then I'm going to just tell you a little bit about some of the other weird things that we looked at. Mary, you can interrupt me at any time here. I don't have to cover these four. I'm going to try to go over them quickly, but do interrupt me if I'm going on too long. So I just want to talk about four kind of cool side studies that we did. One was about predator recognition by native fish. One was about nutrient budgets. One was about the soundscape, how lionfish affect the reef soundscape, and one was about economics. So really quickly, one question for why lionfish were so successful as predators was whether native fish did not recognize them as potential predators. So we approached this question in two different ways. I had two master students, Adrian Berchtold approached this in a conventional way in the laboratory, exposing little native parrotfish to visual cues and odor cues of lionfish and native predators. Lily Haynes took the same question into the field, and here we displaced little damselfish from their home patch, released them at different distances, and saw whether they went straight home or took a big detour if there was a model of a lionfish or a model of a native predator in the way. So you've got the two sets of results, two very different approaches to the very same question, and essentially the same answer. So from the lab experiment, what you have here is at 0, the little parrotfish are right against the partition between themselves and the lionfish or the grouper or the predator, and anything positive means that the 0 is the midpoint of their compartment. Anything closer means they came closer to the partition. Anything negative means they moved away from the partition. And you can see that the little parrotfish reacted to groupers. They moved as far away as they could, but they really did not react very much to lionfish. They stayed put in the same way as they did towards controls. In the field, Lily looked at the extra homing time from a direct line and saw that the groupers did a big arc to get back home around the grouper model, but did not basically beeline home when there was a lionfish model as they did where there was a non-piscivorous grunt model. So these little native fish are not recognizing lionfish as a threat, which is obviously to the advantage of the lionfish. However, Adrian's next step in her experiment revealed something really cool. So what she did was to remove the partition between the lionfish and the parrotfish and let the lionfish hunt the parrotfish. And she looked at the time that the parrotfish survived. I know it sounds kind of mean, but it was an interesting experiment. And what she ended up finding was where the little parrotfish had grown up was really important in determining whether they would survive lionfish predation. So the little parrotfish that came from reefs where lionfish density was low ended up getting eaten very quickly, and very few of them survived. Whereas the little parrotfish that grew up on reefs where lionfish density was high, very few of them got eaten, and most of them survived. We think what's going on here is the same mechanism, but that we talked about before, is that on reefs where lionfish density is high, the bold little parrotfish are eaten, and what's left are pretty shy lionfish that tend to spend a lot of time hiding. And when there's not very many lionfish around, then these bold fish are abundant. And we've got data actually on hiding by these little parrotfish that supports this. So lionfish are essentially creating a pressure that favors the survival of the shyest. I do realize what the time is. Do you want me to carry on? Do you want me to stop?
If we stop in 2 minutes, that would leave us about 10 minutes for questions. Does that sound reasonable?
Yeah, that sounds fine. So I'm not going to tell you about pee, although that's kind of a cool thing that I was really interested in. I'm going to tell you about this one because this one was a real interesting thing. So I invited a postdoc from the UK, Sophie Nedelec, who's a sound specialist. And I really wanted to know whether lionfish were affecting the soundscape of the reef. Now, unfortunately, we tried this before you guys joined, and you won't be able to hear the sound. This is a recording of the sound of a reef. So this is time in along the x-axis and frequency over here. What you would have heard, if you could have heard it, is essentially something that sounds like when you pour milk on Rice Krispies, so a constant snap, crackle, and pop. And the high-pitched snap, crackles, and pops are made by invertebrates, usually snapping shrimp. And you might have heard, if your hearing was really good, some much, much lower grunt-like sounds, which are fish vocalizations. Now, we did this work. I had no idea it would entail working all night, basically from 11 p.m. to 3 in the morning for days and days and days, but that's what we did. And lo and behold, when we analyzed sort of two aspects of sound, so sound pressure level and particle acceleration from the noises coming off these reefs, we discovered that the patches where we had removed lionfish, so these were patches that were part of that same experiment, the patches where there were no lionfish, the sound was louder than the patches where there were lionfish. And the reason why I'm telling you this is that sound from a reef is a really important cue for recruitment of little fish. So it looks like lionfish impose like a double whammy on reefs. They're actually eating the fish and the invertebrates that are creating the sound. So they're creating, they're attenuating sound, so there's actually less recruitment of fish. So two things that is driving down these declines in small fish, small native fish that we documented. So the money thing's also cool. The future, what the future holds, lionfish have already shifted diets. They're eating a lot more invertebrates, a lot less fish, because they've depleted fish so much. We've started to see declines in lionfish abundance in some places, like in the Bahamas, which probably points to these ecosystems sort of starting to reach some sort of equilibrium that now includes lionfish as a mesopredator. The cool thing is that in the Southern Caribbean, we haven't seen these large, huge abundances that we've seen in the Northern Caribbean or the Gulf of Mexico. And we don't know whether that's going to happen in the future or whether that's just never going to happen. And one worry that I have is that we're looking at an invasion debt, whereby lionfish are eating the juveniles of, for example, commercially important fish or long-lived fish, and we're not yet seeing the impacts of this. So thank you very much for attending and for your attention.
Thank you so much, Dr. Cote. That was fantastic. I'm sure there will be many questions. I am going to stop our recording now.